Garren January as a research fellow at the University of Plymouth. So Grant is a research fellow in natural product discovery on the South Africa-UK Antibiotic Accelerator Initiative, working with Professor Mathew Upton of the Antibiotic Resistant Pathogens Research Group at the University of Plymouth. This research project in marine biodiscovery forms part of an exciting initiative between the United Kingdom and South African Medical Research Councils, with the principal aim of setting up a South African-UK Antibiotic Accelerator Initiative or Discovery Hub, particularly investigating South Africa, UK and polar marine environments for novel antimicrobials. Over to you, Grant.
Thank you, and good afternoon, everyone. Thank you for the opportunity to speak. So I'll just introduce my talk, Marine Natural Products as a Source of New and Novel Antimicrobials, Investigating Host and Microbial Symbionts as Producers. And briefly, my talk just has some background information, followed by my workflow for culture-dependent and culture-independent studies, as well as the next steps I'd like to follow in this project. So firstly, why are we interested in the marine environment? Well, we all know that there is high biological diversity, and the deep sea in particular has fluctuations in light, temperature, oxygen pressure, pH, and salinity, and microorganisms have molecular and biochemical adaptations to these extremes. These adaptations include secondary metabolites or natural products that potentially have novel biological activities. And the hypothesis is that by prospecting for these natural products, we could find pharmaceutical compounds of interest. Thus far, we have quite a few marine-derived FDA-approved drugs for treating things such as cancer, chronic pain, antivirals, anti-obesity drugs. There are a few antibacterial antibiotics, but they are in clinical trial phases and have not yet got to the FDA-approved stage, but there is still hope for that. So my culture-dependent workflow started off with getting several marine animals from Antarctica, thanks to a collaboration with the British Antarctic Survey, as well as some local seagrass and seawater from the UK. I would then wash that with artificial seawater, getting rid of transient bacteria and debris, cut some tissue, make some serial dilutions, and then do some pre-treatments if I want to select for things like spore formers or only for gram-positives, for example. I would have at least 8 different media with different carbon and nitrogen sources, with and without antimicrobials, either using agar or gellan gum, having copiotrophic or oligotrophic media, then either with or without sea salt. I'd have 3 different temperatures under standard oxic and pressure environments for at least 4 weeks of cultivation. And that gave me something with over 5,000 plates to start with, so quite a lot of plates to process and colonies to work with. Just a rough summary: from the marine invertebrates, over 120 colonies; from sponges, over 40. I've also got a collection of Upton group producer organisms given to me by previous students that I've left, and then 30 from my seagrass samples. And just below is a little image of some of the morphological diversity you see from the plates I've been getting, the isolates. Right, so once we have the xenic cultures, we then go on to primary screening on solid agar plates using various simultaneous antagonistic assays. We've got things such as cross-streaking, a spot to smear test, soft agar overlay assays, disk diffusion assays, TLC biotography, and wild diffusion assay. I won't go into them, just highlighting them here. So once we've got an isolate that is active, we then put them into liquid media using a one-strain mini compounds or OSMAC framework. And this is basically the ability of a single strain to produce different molecules when grown under different environmental conditions. You have silent biosynthetic gene classes, which are not typically expressed under lab conditions. And by triggering the expression of these BGCs, we could unlock the chemical diversity, allowing for the discovery of new molecules of interest. This can be done by changing things such as the temperature, the time of incubation, co-cultivation with neighboring isolates, aeration, so oxic and anoxic conditions, having different nutrient regimes, so carbon, nitrogen, sulfur, and phosphorus sources within your cultivation media. Just to show you a quick case study of two of my strains, I've got OC14, which is a isolate from an octocoryl, and then SP20A, an isolate from a sponge, an Arctic sponge. In the front row, you see SP20A grown in several different media for antibiotic production of actinobacteria. And you can see just by changing things as the carbon and nitrogen source, you have different expression profiles showing different phenotypes in this liquid media. So why was I interested in these two strains? Well, the first one, Streptomyces von Leahy, there's only two known genomes on NCBI GenBank, one partial, one complete, and it's only known to produce an antifungal. This is the first time, to the best of our knowledge, that it's been discovered from an octocoryl or from Antarctica, so it was something of interest. Likewise, with the Micromonospora SP20A, previously been isolated from an array of different environments. Again, the first time from Antarctica, to the best of our knowledge, also known to produce anti-tumor antibiotics. So it was of interest to us because of that. So I did a little extraction of these cultures, tested it on a disk diffusion assay. We'd look for a zone of inhibition and then measure the size of the diameter, the zone of clearance. 6 millimeters is the size of the disc, so that has to be subtracted from this diameter readings. So OC14 with 4 different types of media had prominent activity against Micrococcus luteus, which is a gram-positive bacterium. So just for the purpose of this talk, I'll just speak about OC14. So grew it in its optimal medium up to stationary phase, centrifuged that culture, did a liquid-liquid extraction with ethyl acetate, splitting it into an organic solvent and an aqueous phase. I would dry that down or concentrate it and then test for activity on disk diffusion or agar diffusion assays. And we could see that the aqueous phase showed anti-gram positive activity against Micrococcus luteus, so that was good to see. We then thought about fractionating this to concentrate activity, so using a reverse phase C18 SPE column. We put 45 milliliters of the active aqueous phase through the column, eluting with a decreasing solvent polarity, so from water up into 100% methanol. Sampled out an aliquot, concentrated that down, again, made spread plates with pathogens, and then tested for activity. And we could see activity was retained from the 25% methanol SPE fraction, again, against Micrococcus luteus. So we went on to partial purification. So we took this 25% fraction, dried it down in a rotary evaporator. We got 160 milligrams of dried mass, took 45 milligrams of that, put it onto an isolator 1 instrument, which is just flash chromatography, again, using a reverse phase C18 column, eluting with a decreasing solvent polarity from water to acetonitrile. And we see these 2 peaks of interest in blue and yellow, and then the tailing section in orange at the right of the second peak and the green section to the left. So all 4 of those peaks or areas were dried down and tested for activity. And we see activity from several of the peaks or areas against Micrococcus, but then also extending the activity slightly to Staphylococcus aureus. And also, the green area on the chromatogram, or at least that fraction, showed activity against E. coli. So the spectrum of activity was even extended from not only gram-positive, but also to gram-negative as well. So we also are interested in genome mining of these isolates to see what the biosynthetic potential is. So we decided to do Illumina short-read sequencing, as well as Oxford Nanopores min-iron long-read sequencing, doing data curation, combining that to make a hybrid genome assembly. So we have a nice, almost complete genome, which we can give us a more robust annotation. Right, so at the moment, we only have the Illumina data, but that's okay. I took the annotated genome, put it through anti-smash, which basically mines the genome for biosynthetic gene clusters. And using these thresholds established by a very recent paper by Benound et al. In 2021, looking at Antarctic BGCs and their biosynthetic potential, we have highlighted several BGCs with low enough similarity that it could potentially be good targets for novel compound biosynthesis, but I won't go into that because of time. So these several clusters highlighted in yellow have the potential to produce these antibiotic compounds, so we will be mining that further in the future. Okay, So the next steps I'd like to employ is purify my active metabolites, as well as using LC-MS metabolite dereplication using molecular networking, use the genome mining data to inform culturing parameters, do secondary screening of the purified compounds using microdilution baths, continue building and screening the marine microbial culture collection for activity, and then also doing solid liquid extractions from the host organism itself and not just from the microbial symbiont cultures. With that, I'd just like to acknowledge the following people and organizations for their support and funding, and thank you for your attention.